It's kind of sparsely populated out here today. Everybody decided to take Friday off? Maybe I should have taken Friday off too. So, I'm happy to report the exams are finally graded. Yes. Now, a couple things about that. Yep, just a second. A couple things about that. First of all, they will be available for pickup starting at 2 o'clock this afternoon. Okay? Not before then. So you go racing out of here to go get it. They're going to tell you no, and they're probably going to take your name and let me know that you came early, because it bothers the office people. So please, I'm going to tell you where to get it, but you can line up, but don't go bugging people until 2 o'clock, okay? Please. All right, so it'll be available in the Biochem office, ALS 2011, starting at 2 o'clock. They will be there handling exams until 4, okay? So if you, oh my God, I've got class from 2 to 4, et cetera, et cetera. I should be around after 4, so if you didn't get in that 2 to 4 time window, come see me, and I will, if I'm there, obviously go dig up your exam for you, okay? So I'm sorry it took so long. It doesn't usually take that long, but in any event, the exam was graded, and I was overall pleased with performance. Of course, there's always people who can do better, and I'm happy to help people to do better. So if that's you, come see me. I'll be happy to meet with you. The TAs are also available to help you if that happens. The low score on the exam was 9. The high score on the exam achieved by 2 or 3 people, I can't remember how many exactly, was 99. Okay. And the overall average, which is what I look at in terms of performance, was 66.5. And so my feeling is if that an exam has an average between about 65 and 70, it's a pretty good measure of things, and it's a pretty good performance. And so that's where things were, okay? This evening I will post a curve that will show you where your letter grade fits into all that, and I'll put that on Canvas. And actually what I'll do is, I'll post that on the web. I will post that on the web. I'll probably also put it on Canvas for that matter. But as you know, the web is what I prefer to use for most of the stuff that we'll do. If you have questions about your exam or anything, let me know. There is a key posted outside of my office. And my office is ALS 2145 if you don't already have that. And so please feel free to look at the key. If you read, you should read the syllabus. Make sure you understand the rules about exams and so forth I see people shaking their heads. They read the syllabus. They know what I'm talking about. So please read the syllabus regarding exams. Okay, so we are ready now to move our attention to talking about the last of our metabolic pathways, and this concerns nucleotide metabolism. So nucleotide metabolism, in many ways, I think is the most interesting of all the metabolic pathways. And the reason for that is because of the really intricate system of regulation that's involved in controlling how much of any given nucleotide is made. Okay? We're going to see balances in several systems for balancing relative amounts of different nucleotides that the cell uses. And there's a very important reason why the cell does this. Why do cells care so much about the relative amounts of adenine versus guanine nucleotides, or cytosine versus guanine, or cytosine versus thymine, or any of those? And the answer to that question is because imbalances in the levels of nucleotides, too much of one, too little of another, lead to mutation. Cells avoid mutation as much as they can, okay? Because mutation, of course, can be quite detrimental to the organism. Mutation is also, of course, how it is that evolution happens. So there is no perfect system that prevents mutation. Some mutations are, in fact, beneficial. Most mutations are detrimental. So cells are pretty protective about mutations. So consequently, the cell balances nucleotides in levels that will reduce the level of mutation. And that's why this balance is so important. Okay, well, a lot of this is basic knowledge that you already have. You know, of course, that nucleic acids have as their building blocks nucleotides. You know that nucleic acids include the ribonucleic acids, which are RNA, and the deoxyribonucleic acids, which, of course, are DNA. You know that RNA contains the nucleotides with the bases uracil, cytosine, guanine, and adenine. Now, I just gave you the bases. Please don't confuse the names of the bases with the names of the nucleosides or the nucleotides. So if I talk about a base, I'm talking about cytosine, for example. If I'm talking about a nucleoside, I'm talking about cytidine. Okay? Well, what's the difference between a base and a nucleoside? Right? A base is, as you've learned in basic biology, the things that comprise the information in the RNA or DNA. Okay? The five bases that we see are adenine, guanine, cytosine, uracil, and thymine. Thymine, of course, replacing uracil in the DNA compared to RNA. Okay? A nucleoside is something that contains a sugar and a base. Okay? So if I say something is a nucleoside, I mean it contains a sugar and a base. The sugar is ribose in the case of RNA, deoxyribose in the case of DNA. We'll see how deoxyribose is made later. Okay? A nucleotide is something that contains a base, a sugar, and at least one phosphate. So adenosine monophosphate is a nucleotide because it contains one phosphate. Adenosine triphosphate is a nucleotide because it contains three phosphates. So as long as it has at least one phosphate, it is a nucleotide. Okay? A nucleoside monophosphate, by definition, then, is a nucleotide because it's a nucleoside that contains a phosphate. Make sense? Okay, so don't confuse those terms, base, nucleoside, and nucleotide. The bases names are all shown right here. If we want nucleoside names, we talk about adenosine. That would mean that it would contain adenine attached to a sugar. Guanosine, which would have guanine attached to a sugar. Cytidine, which contains cytosine attached to a sugar. Uridine, which is uracil attached to a sugar. And finally, thymidine, which is thymine attached to a sugar. Okay? So those are the names that we will use in the class. And, question? Okay. So those are important. We will see as we go through this that there are a couple considerations for making nucleotides. We'll see that the ribonucleotides are the nucleotides that are made first, and we think of order. Okay? Ribonucleotides are made first, and they can be made by two pathways. One we call de novo, which means literally making it from scratch. And we'll see that the scratch is pretty simple looking molecules that are used to make nucleosides and nucleotides. And the other is by salvage. And salvage is like a salvage yard itself. You go to a salvage yard to buy a starter off of a car that has been driven for many years because that's cheaper. It's cheaper to go buy that starter off of a used car than it is to go buy a brand new starter. And it's similarly easier or less energetically expensive for a cell to make a nucleotide from pieces of a nucleotide. Okay, so that's the salvage pathway. I won't say too much about salvage pathways. I'll talk about them relative to genetic disease later, but I won't say a lot about those. Most of what I will talk about are the de novo, or the pathways that work from scratch. Purines are made by one pathway, pyrmidines are made by another pathway, and deoxyribonucleotides are made from the ribonucleotides. They're made specifically from ribonucleoside diphosphates, like ADP, UDP, CDP, GDP. Those are the starting materials for making the deoxyribonucleotides. Thymidine nucleotides are made by an unusual pathway that's actually separate from the others that I'll talk about, and it's made from uridine nucleotides. Not a big surprise there. But thymidine metabolism is separate and interesting. Okay, now, I talked about, yes, question?
So a nucleotide is a nucleoside triphosphate?
A nucleotide is a nucleoside with at least one phosphate. So anything that has at least one phosphate on a nucleoside is a nucleotide. Okay? Other questions? Okay. Well, I said that nucleotides, nucleosides, for our purposes of the moment, they're not that distinct from each other in terms of synthesis. The molecules are made from very simple starting components. So this shows the starting components for the bases, whether they're a purine, as shown on the left, or a pyrimidine, as shown on the right. And each individual atom that's contained within these rings is identified in terms of its source. And as you look at this, it's interesting. Look at, first to start at purines. The central part of purines comes from glycine. So glycine is glycine is an important precursor in the synthesis of purine nucleotides. Aspartic acid contributes the nitrogen up there. Now you're not going to memorize this. I'm not giving you this so that you look and say, okay, Kevin said which one came from where. That's not what I'm going to ask you. But I want to impress on you the link between the synthesis of the bases of the nucleosides and nucleotides and amino acids. Look at aspartate, look at glutamine, aspartate, glycine, okay? So bases aren't something that are overly exotic, and we know, excuse me, that the chemistry that existed on ancient Earth during the time that life was evolving contained molecules like this that we now know can come together and make structures that gave rise to the nucleic acids, okay? It didn't take exotic chemistry, and it doesn't take exotic chemistry for these things to happen and for these kinds of molecules to form. Now, there are other things that are here, and these are later evolutionary events, but these involve what we call formate metabolism, and this is single carbon metabolism. And I'll say a little bit about that because the folate, I'm saying formate, it's not formate, folate, F-O-L-A-T-E, folate metabolism is very important for the synthesis of the purines in particular. And as I say, we'll talk more about that as we get into those. Okay, now, one of the things that you're going to see as I go through and talk about these pathways is that there's a lot of information. Now, I could become your worst enemy by saying, okay, memorize all these structures, all these names, all these enzymes, and all I would have accomplished is you would have gotten frustrated and mad at me, and you would forget it as soon as the exam was over. So there's no reason for me to ask you to memorize those things, okay? What I will do as I go through these is point out some basic features, and as always, my guideline is what I talk about in class is what you're responsible for, okay? So if I don't talk about, I'm not going to say it because if I say it then you become responsible for it. If I don't talk about some of these things, right, then you're not responsible for them. But you can see looking through here, various molecules and things coming in. You can see input of energy. You can see input of nitrogens. Here's where glycine comes in, okay? And those are the bigger picture things. So I'm going to give you a big picture thing right now. We're going to start the discussion here with the synthesis of the purine nucleotides, okay? The purine nucleotides are distinct in the way that their reactions go, okay? Purine nucleotides are made by starting with ribose 5-phosphate. Now, ribose 5-phosphate, you learned last term, is made in the pentose phosphate pathway. The pentose phosphate pathway is therefore critical for the synthesis of purine nucleotides, okay? It's also critical for the synthesis of pyrmidine nucleotides, but for a different reason, or in a different way, okay? So ribose 5-phosphate is a critical molecule in this overall process. It's a starting molecule for the synthesis of the purines. If you look at ribose 5-phosphate, its structure is shown there, and that's pretty straightforward. You notice that this phosphate that's right here, okay, doesn't have anything attached to it, and further, carbon number 1, which is right there, is a hydroxyl, and it's on carbon number 1 that the base will actually be built. Okay, the base will be built. And that's the primary difference in the overall scheme of the synthesis of purines versus pyrimidines. That is, the bases are built on ribose, or ribose 5-phosphate, okay, in purines. They're not built on there in pyrimidines. Okay, they're attached later. There's a fundamental difference between them. Another common feature, or at least a common feature, between the synthesis of pyrimidine nucleotides and purine nucleotides is the involvement of this molecule right here. It's called PRPP. PRPP stands for phosphoribosylphosphate pyrophosphate. You don't need to know that name, but you can call it PRPP. Phosphoribosylphosphate pyrophosphate, again, the name tells us something about what it is. Phosphoribosyl, there's the phospho, there's the ribosyl. Pyrophosphate, there's the pyrophosphate. So we've got phosphoribosyl pyrophosphate. It's basically a ribose that has one phosphate on one end and two phosphates on the other end. That molecule is involved in both the synthesis of purine and pyrimidine nucleotides. Okay. We go through here and we look at the synthesis of these molecules, and we can see we start looking at the ring, but then we start following what's actually with the sugar ring, but then we just focus our attention onto the bases because it starts getting complicated. So let me show you these reactions in another way. Oh, actually, before I do that, the overall scheme here is important. Okay. The overall scheme in the synthesis of purine nucleotides is that adenine nucleotides and guanine nucleotides both follow the same path until they get up here to this molecule called IMP. You can know it as IMP. I'll tell you it's inosine monophosphate, which is a purine nucleotide. And we'll actually see that nucleotide appear in nucleic acids later. It appears in transfer RNAs many times, and we'll talk about that later. But IMP is a branch molecule. One side goes to the synthesis of the adenine-containing nucleotides, and the other goes to the synthesis of the guanine-containing nucleotides. Okay? So IMP is the branch. Up until IMP, everything is the same. And if you look at this scheme I've shown here, there are 10 steps that get you to IMP. All right. Well, as I said, I used some abbreviation to fit all that stuff on there. So let's take a look at some of the relevant, important steps along the way. This figure is important because it shows two of the important enzymes, and really two of the enzymes that I think you should know the names of. Okay? And the reason I want you to know the names of these enzymes is because of their roles in synthesis. Okay? Roles in synthesis. PRPP aminotransferase is the more important of the two. Okay? It catalyzes the second reaction in this process. The first one is just involved in making PRPP. And PRPP, of course, as I say, we know is important for both purines and pyrimidines. PRPP aminotransferase has an unusual regulation. Okay? So let's take a look at its regulation. All right? So we're going to look at the regulation of the PRPP synthetase. Okay? Oh, that's the reactions. Okay. Now, that enzyme's regulation is described right here. Okay, that's where that blank arrow is there, PRPP aminotransferase. PRPP aminotransferase is the primary regulated enzyme in purine de novo biosynthesis. I'll repeat that. PRPP aminotransferase is the primary regulated enzyme in purine de novo biosynthesis. Now, I said its regulation is unusual, and its regulation is really interesting. Okay? The enzyme is regulated in two ways. One way turns it off. And how does this enzyme get turned off? It gets turned off, okay, if both AMP and GMP bind to it. So these are feedback inhibitors of the enzyme. They're both negative regulators of the enzyme. But it takes both of them to bind to the enzyme to turn the enzyme off. If only one of them binds, the enzyme is slowed down. I call it partially inhibited. It's slowed down by the binding of one of them. Now, we haven't talked about a regulation scheme like that previously. And this is one of the few enzymes that actually has this regulation scheme. Why does it have this scheme? The reason it has this scheme is for the purpose of balance. Okay? First of all, the balance achieved by the negative regulation of both means that if we have too many AMPs and GMPs, of course these are purine nucleotides, if we have too many purine nucleotides, the entire pathway is shut off. We don't want to have too many purines, right? If we have too many purines, then we don't have enough pyrimidines, and we've got a problem. So this helps to control the synthesis of purines overall. This enzyme also helps to balance the relative amounts of the adenine versus the guanine nucleotides. How does it do that? Well, let's imagine I've got too much AMP in the cell. I don't have enough GMP in the cell. What would happen? I have too much AMP, only AMP is going to bind to the enzyme. I have too little GMP. What's going to happen if I have too little GMP? If AMP completely turned off the enzyme, I wouldn't be able to make GMP because they're both done by the same pathway. By slowing the enzyme down, okay, I favor the continuation of this pathway. Now I'm going to show you later that AMP synthesis will turn off itself in another place. But it's important that we keep this enzyme partly active so we can make guanine nucleotides if we have too much AMP and not enough GMP. Or vice versa. If I have too much GMP and not enough AMP, I want to keep the pathway going so I can make AMP. Make sense? I'm not going to tell another joke today. You guys look like you need it, though. Yes, question?
If you slow it down, would it still make the AMP and GMP equal amounts?
Okay, so good question. If I slow it down, will it still make AMP and GMP in equal amounts? As we will see, GMP will turn off its own synthesis, and AMP will turn off its own synthesis. So no, it won't. But this is, and it's a very good question, okay? This is why it's very important that there be multiple ways of balancing things. Because if I didn't have multiple ways of balancing things, even though I slowed the enzyme down, I would still make equal amounts of the two, and I would still have the imbalance, right? Make sense? Okay. So the regulation of this enzyme is really, really critical for the overall process. Okay, I promised I was going to show you the steps. So there's a little bit more detail in the steps. And again, you're not going to memorize those structures. But with this figure, I like it a little bit better because you can start to see the synthesis of that purine ring, or the purine double ring, as we go through this process. Okay? We start right here with, this is the PRPP aminotransferase enzyme right here. And this ring structure starts to grow. All of a sudden we've closed the ring, we start building the second ring, and finally we get over here where we've just about closed the ring. And the next step actually will close the overall ring. Okay? Okay. Up here you can see that we have gotten to IMP. We've closed the ring. IMP is a purine nucleotide. We don't use it, none of the polymerases use it to put into nucleic acids. It gets into transfer RNA through a chemical modification. So it's a purine-like ring, but it's not really what we would call a purine nucleotide, even though it has a sort of a purine ring associated with it. The important point about IMP, though, is the fact that it's a branch point for the synthesis, as I noted, of the guanine nucleotides and also the adenine nucleotides. Okay? Well, this is a very interesting setup here, and the setup that you see here also helps to provide that balance in the synthesis of each one. Let's follow the top pathway first. We go from IMP to this molecule called XMP, and no, I don't care if you memorize the name XMP and all that. If you know that IMP is going to guanine nucleotides, you're in good shape. And if you know that it goes from GMP to GDP to GTP, you're in good shape. That's pretty straightforward. You put one phosphate on, then you put another phosphate on, then you put another phosphate on, and you got GTP. Okay? This enzyme shown here, excuse me, IMP dehydrogenase, is inhibited by GMP. It's inhibited by GMP. So GMP regulates its own synthesis. If there's too much GMP, this enzyme will be turned off. Now think about the PRPP aminotransferase regulation. If I had too much GMP and not enough AMP, I would keep the PRPP aminotransferase slightly active, and when we got to the IMP reaction, GMP would turn off its own synthesis so that everything that's made would become adenine nucleotides. Another interesting thing about this pathway is if we look right here, okay? Notice the energy that's used to make GMP nucleotides. It's ATP. Well, because if I didn't have enough guanine nucleotides, I wouldn't be able to use guanine nucleotides to synthesize guanine nucleotides, right? So it's appropriate that I have adenine nucleotides used to make guanine nucleotides. Adenine nucleotides are the energy source. If we look down here, we see that guanine nucleotides are the energy source for making adenine nucleotides. These are also balancing things. They're kind of necessary too if we're short of one or the other, right? On the bottom pathway leading to the adenine nucleotides, we see this similar thing. We see the synthesis of the monophosphate, the diphosphate, and the triphosphate. Okay? And this enzyme, adenylosuccinate synthetase, and yes, that name's important, just like the top IMP dehydrogenase name was important. Okay? Adenylosuccinate synthetase is inhibited by, what do you suppose? Our friend AMP. So AMP inhibits its own synthesis just like GMP inhibits its own synthesis. And as a result of the coordinated action of PRPP aminotransferase and these two enzymes, the cell makes the relatively appropriate ratio of guanine and adenine nucleotides. Okay, kind of cool. Now there's a couple other enzymes that are on here that are important, and they're actually very easy to learn. These are the two most complicated enzyme names that you'll have, okay? GMP kinase. What does a kinase do? Puts phosphates on. So a GMP kinase puts phosphates onto GMP, and when you put a phosphate onto GMP, you get GDP. Right? AMP kinase, or in this case, adenylate kinase, they're equivalent names. You can call it AMP kinase if you wish, okay? Adenylate kinase takes and puts a phosphate onto adenylate. AMP is also known as adenylate. Okay? Makes ADP. So we have specific mononucleoside phosphate kinases. That is, there's a specific one for GMP, there's a specific one for AMP. Now I point that out because when we get to the diphosphates, we don't have specific ones. The same enzyme is used to convert all of the nucleoside diphosphates into all of the nucleoside triphosphates. It's called NDPK, nucleoside diphosphokinase. Again, the name tells you, right? Nucleoside diphospho, it's a nucleoside, it's got two phosphates on it. Kinase puts a phosphate onto it. You're welcome to call it NDPK. That's what I always call it. NDPK catalyzes the conversion of all of the diphosphates into all of the triphosphates. UDP goes to UTP as a result of NDPK. CDP goes to CTP as a result of NDPK. TDP goes to TTP as a result of NTPK. DADP goes to DATP, that's the deoxy forms, as a result of NTPK. So you know one enzyme, you know all of the reactions for that. I've told you that there are specific monophosphokinase. What do you suppose we would call the monokinase that converts CMP into CDP? CMP kinase, right? So they're very easy to learn. Very, very easy to learn. Okay, so at this point, we've gone through these reactions and we have made triphosphates. Triphosphates are essential because if we're going to make nucleic acids, the polymerases use triphosphates as the starting material. Okay, we'll talk about DNA replication and transcription in the weeks to come, but having triphosphates is necessary to do that. Notice that what we've done here is all the ones have, these are all ribonucleotides that we're making. We haven't talked about deoxyribonucleotides. When I talk about the deoxys, I'll put a D in front of there. If I said DGTP, I'm talking about deoxyguanosine triphosphate. So I haven't made any deoxys yet. I've only made the ribo forms. Yes?
Now, because high concentration of AMP is linked to a state of low energy, would a state of high energy, like a resting state, be favorable to mutations? Or are there a bunch of them?
Oh, that's a very good question. So his question is, I've said that the regulation's happening at the monophosphate level. Does that mean that if we have a high energy state where we have a lot of ATP, that would be more mutationally favorable? Am I stating your question correctly? It's a very good thought, okay? And the answer is generally no, okay? Because along the way, that synthesis of this has been slowed down, stopped as appropriate. But you're correct. If we sat there for a long time with this guy sitting in the ATP level, and we had very little at this level, then we would in fact have an issue. But we tend to think about these in terms of this is high energy, therefore we don't have any of this, and that's not true. So it's the relative amounts. That's a very, very good question. Yes, question here.
So in the ATP, you use in the ATP pathway, and then on the GTP pathway, they use ATP?
Yes.
Is it the same ATP or?
Okay, so the question is the ATP that's used here this ATP? And the answer is no. You got billions of these in the cell. Billions of these. So we tend to like to look at these and say, okay, well, there's that one, so it goes back over here, and there's that one, so it goes back over here. But that's not what happens. Is that your question? Okay. Yeah. Good. You guys are thinking about this stuff. All right. So let's summarize. Okay. So what up there have I not said? Purines are made de novo. PRPP is made as a starting material. These are simple compounds, branch point, I think everything I've already said. So that's just my checklist to make sure I haven't forgotten anything by there. All right, let's turn our attention to pyrimidine biosynthesis. And again, we're talking about the de novo pathway. I didn't talk about the salvage and the purines. I'll talk about that later. And I haven't talked about salvage of the pyrimidines either. We're looking at the de novo. The de novo pathway starts from scratch. And this is an overview of the pathway. And we're going to treat this pathway very similarly to the way that we did the purine pathway. But there are differences, okay? One of the differences is we start making the base before we attach it to the sugar. So for pyrimidines, we make the base first, okay? And then after we've made the base, we start putting it onto a sugar and then making the nucleotides, okay? Look at the starting materials right here, these very simple starting materials that we start with to make pyrimidines. Bicarbonate, glutamine, ATP, and water. Cells have plenty of this stuff sitting around, and we go through a series of reactions. We're not going to follow the individual reactions, okay? One of the things we don't see in the synthesis of pyrimidines, we don't see that branch point that we saw in the purines, okay? There's not a branch point. That means that the regulation here is a little bit different, and there are some considerations. The regulation to balance these is different than it is for the purines, okay? Now, let's look at that regulation. Oh, and by the way, one of the things I should point out is that the ring is built first, but the molecule that the ring becomes attached to is the PRPP molecule. Okay? So PRPP is what it gets attached to, and that provides the source of the ribose for the ribonucleotide. All right. Well, let's, actually I'll come back to that. Let's talk about the regulation again. Here's the overall pathway, starting with these very simple precursors, and ending up, the end of the pathway is the synthesis of cytidine triphosphate, CTP. Okay? That's the end of the pathway. The first pyrimidine nucleotide that we make is right here, UMP, uridine monophosphate. Okay? Uridine monophosphate is phosphorylated by UMP kinase to make UDP. And UDP is phosphorylated by what to make UTP? NDPK, right? So we go from here, 2 steps, we're at UTP. UTP is converted into CTP by this enzyme here. It's called CTP synthase. Very simple name, CTP synthase. There are 3 important regulations in this pathway that we need to consider. One of these you've already learned. You learned it last term, and I've mentioned it briefly already this term. It's the enzyme known as aspartate transcarbamylase, or ATCase. Okay? The name here, aspartate transcarbamylase, all right? ATCase is what you're more likely to call it. You may remember from last term that ATCase is one of the prime enzymes we talk about allosteric regulation with. And to refresh your memory from last term, ATCase is allosterically regulated, okay, by 3 different things. Okay? 3 different things. Okay? Let me see if I've got, I can't remember if I've got a figure for that as not, or not. Okay, so regulation of aspartate transcarbamylase is as follows, all right? That the reaction it's catalyzing is the synthesis of this molecule. That's where it gets its name. You don't need to worry about that, okay? But the enzyme is controlled by 3 different things. ATP activates the enzyme. What's an ATP? What's ATP an indicator of? High energy. What else is it an indicator of? What? It's an indicator of high energy, but what else is it an indicator of? High purines, yes. Okay? So if we have a lot of ATP, we have a lot of purines, right? And so if I have a lot of purines, do I want to have a lot of pyrimidines? Absolutely. If I have a lot of energy, do I want to be making pyrimidines? Probably do, right? Because if I have a lot of energy, the cell gets to go make whoopee, which means it gets to divide. And to divide, what does it need? It needs nucleotides, right? So ATP is a very important positive activator of the enzyme, or positive regulator of the enzyme. CTP is an inhibitor of the enzyme. What's CTP an indicator of? Pyrimidines, right? We don't want to be making a lot of pyrimidines if we have plenty of pyrimidines. And so this is a feedback inhibitor of the enzyme. The third one's kind of cool. Aspartate, aspartic acid, okay, is an activator of the enzyme. What's aspartate an indicator of? What is aspartate? What's that? It's an amino acid. So it's an indicator of amino acids. And what are amino acids important for? Synthesis of proteins, right? And what do cells need to make if they're going to divide? Proteins. So just like an indicator of high energy is important, so too is the high indicator of amino acids for protein synthesis important. Both aspartate and ATP will activate the enzyme. CTP will inhibit the enzyme. This means that this enzyme is playing a very important role in the balancing of the relative amounts of purines versus pyrimidines. Question here. Is there a reason why aspartate is the only amino acid? No, it's just the way it evolved. But aspartate is central to numerous things, and aspartate is readily made from oxaloacetate, which is in the citric acid cycle, and so now we start seeing connections to energy in other ways. So that's probably why, but beyond that I can't tell you. Was there a question here? Yeah. Right here first.
If we're trying to balance purines and pyrimidines, how do some organisms have more cytosine and guanine in really harsh environments?
So the question is, if this is balancing purines and pyrimidines, how do some organisms have high levels of cytosine, guanine in harsh environments? That's a different kind of a regulation, and usually for cells that are in unusual environments or that have unusual genomes, there's other regulatory things that are there. In my postdoctoral work, for example, I worked on an organism called Dictyostelium discoideum, which most people don't know that name, but you've heard of slime molds, and that's what they are. And slime molds, if you look at their DNA, it's bizarre. It's 90% A and T. So in an organism that has those imbalances, they also have to reflect that in their synthetic pathways, and that's getting beyond what we can talk about here. Was there a comment here?
So do you need both aspartate and ATP to activate, or is it just...
Good question. Do you need both aspartate and ATP to activate? No. Either will do it. Either will do it. Generally, if you have plenty of amino acids, you're probably at a pretty good energy state anyway because you wouldn't have gotten to there without it. But yeah, it's an important consideration. But when I talk about balance, think about this. High purines, okay, turns off its own synthesis. High pyrimidines turns off its own synthesis. High purines activates pyrimidine synthesis. So this balance works really well through this one enzyme. Well, one thing I haven't told you is I haven't told you the relative balance of pyrimidines. What's the difference between UTP and CTP, for example? I did tell you that thymidine was in a different territory, so we're not going to worry about its balance because its balance turns out to be controlled by the balance of the uridine nucleotides. So when we think of uridine, we can think of thymidine in the same sense. So if we balance the uridine nucleotides and we balance the CTP, or the cytidine nucleotides, then we will also balance the thymidine nucleotides. So the question is, how do we balance CTP versus UTP? And that turns out to come from the CTP synthetase. And if I go back to that, you can see that, oh, it's not here, sorry. You can see that right here, okay? Now, this enzyme is regulated in a complex fashion. We're not going to worry about the phosphorylation, although the phosphorylation of that enzyme is interesting. This enzyme is activated by GTP. Why do you suppose that's important? What's that? G's and C's are paired, right? High G's better have plenty of C's, right? So that's going to activate it. CTP inhibits its own synthesis. So if CTP levels get too high, it's going to turn off this enzyme, and when it turns off this enzyme, it's going to back up, and what is the precursor used to make CTP? It's UTP. So if I block the enzyme, I'm going to increase the concentration of UTP. So CTP synthetase is the enzyme that balances UTP versus CTP. It balances UTP versus CTP. So now we've got balances between the adenine versus the guanine nucleotides through PRPP aminotransferase. We've got balances between the purines and the pyrimidines through the ATCase. And we've got balances between the CTP and the UTP through the CTP synthetase. When we get to the deoxyribonucleotides, we're going to see that there's some really interesting regulation that occurs there, and all of that regulation occurs through one enzyme. And as you might guess, that turns out to be complicated. We're not going to talk about that today, though, because today is Friday, and I thought you might want to finish a little early, so that's what I'm going to do with you. If we get to sing a song. This is a song that I should have sung a couple of days ago, but I think you'll enjoy. It's called To Make a Cholesterol. Built with acetyl-CoAs are joined together partly thanks to thiolase. They come together 1, 2, 3, 6 carbons known as HMG, and you're on your way to make a cholesterol. Synthesize a mevalonate in the cell requires reducing HMG-CoA as well. The enzyme is a reductase controlled in allosteric ways when the cell's impelled to make a cholesterol. Valonate made in metabolic schemes gets decarboxylated down to isoprenes. They're linked together willy-nilly to build a pp-geranyl in the cell's routines to make a cholesterol. Go step links farnesils, but that's not all. The squalene rearranges to lanosterol. From that there's 19 steps to go before the sterols apropos, which you must recall to make a cholesterol. The regulation of the scheme's complex in ways inhibited by feedback of the reductase. And statins mimic, so they say, the lake of HMG-CoA. So we sing their praise and not make cholesterol. Okay, guys, see you later.